Hey everyone, Dustin Slovene here. This is your Module 5 lecture, Biochemical ID of Gram-positives. Here's an overview of some of the topics we'll be discussing today. This will include some of the biochemicals used to identify gram-positives, including catalase, opticin, bacitracin, PYR, CAMP test. And additionally, we'll be discussing Lancefield antigens and common causes of pharyngitis. Catalase is one of the most important tests used for the differentiation of gram-positives in the clinical laboratory. The catalase test detects the enzyme catalase, which breaks down hydrogen peroxide into water and oxygen. When hydrogen peroxide is added to a colony, rapid bubbling indicates a positive result. An absence of bubbling indicates negative. Staphylococcus and Micrococcus are catalase positive, while Streptococcus and Enterococcus are catalase negative. While Aerococcus are generally catalase negative, catalase can help differentiate Aerococcus urinae, which is catalase positive, from Aerococcus viridans and Aerococcus sanguinicola. Furthermore, it can differentiate Campylobacter fetus, Campylobacter jejuni, and Campylobacter coli from other Campylobacter species. Some organisms, such as certain Enterococci or aerotolerant organisms, may produce a weak bubbling due to peroxidase activity. This is not true catalase. Because Micrococcus and Staphylococcus are both catalase positive, they can be further differentiated by using modified oxidase test. Opticin, also known as P-disc or ethyl hydrocuprin hydrochloride, is an important test for the differentiation of alpha streptococci from pneumococci. A pure isolate of an alpha strep is struck out on a blood agar plate, and the P-disc is embedded onto the blood agar plate. After incubation, the zone of inhibition is measured. A zone that is greater than 14 millimeters is considered positive and indicates that the organism is a pneumococci. If the zone is between 8 and 13 millimeters, this should be confirmed by bile solubility, as this result is considered equivocal. This is the mechanism in which the P-disc works. Other streptococcal species possess differing amino acid sequences in their ATP-C gene, which results in a lack of affinity in non-pneumococcal species. The chemical alters surface tension of pneumococci, inducing lysis. This test must be incubated in 5% CO2, and it should be noted that streptococci and other fastidious organisms in general prefer 5% CO2 incubation. Streptococcus pseudopneumoniae produces a zone size greater than 14 millimeters in ambient air, but less than 14 millimeters when incubated in CO2. This organism has been implicated in COPD and respiratory infections, but its clinical significance remains uncertain. Bacitracin, or A-disc, is used for the presumptive identification of Streptococcus pyogenes, also known as Group A beta strep. A visible zone size, regardless of size, indicates susceptibility, which is an important characteristic of strep pyogenes. Resistant organisms do not possess Lancefield antigen A. Occasionally, some Group C and G strep are bacitracin susceptible, but for purposes of this class and for the BOC, you'll just want to know that Group A strep is susceptible. Additionally, Group A strep are beta hemolytic and PYR positive. The PYR test detects the presence of pyrrolidonyl arylamidase. Organisms with the enzyme hydrolyze PYR and release beta-naphthylamine, which reacts with the developer and results in a cherry red color. Streptococcus pyogenes, Enterococcus species, and Staphylococcus lugdunensis are PYR positive. Now let's move on to the CAMP test, a classic and very testable method for identifying group B streptococci. The CAMP test is used primarily for identification of Streptococcus agalactiae, which is Lancefield group B. Here's the principle. A known strain of Staphylococcus aureus is struck down the center of a sheep blood agar plate. Staph aureus produces beta-lysine, which causes incomplete hemolysis of red blood cells. When a suspected group B streptococcus is struck perpendicular to, but not touching, the Staph aureus streak, something important happens. Streptococcus agalactiae produces a diffusible extracellular protein called CAMP factor. When CAMP factor interacts with the beta-lysine produced by Staph aureus, there's a synergistic enhancement of hemolysis. The result is a very characteristic arrowhead-shaped zone of increased hemolysis at the junction where the two organisms' factors meet. The arrowhead pattern is considered a positive CAMP test. It should be noted that, rather than being an acronym describing function, CAMP is an eponym based on the discoverers' names, Christie, Atkins, and Munch Peterson. So like I mentioned in the previous slide, when doing the CAMP test, Group B exhibits arrowhead-type hemolysis. Clinically, this matters because Group B strep is a major pathogen implicated in neonatal sepsis and meningitis, and historically the CAMP test was used in OB screening around 36 to 37 weeks gestation. Modern labs have shifted to using PCR platforms such as the Cepheid test, so you may see this less often in real practice, although it's still a very testable characteristic. There are other organisms that are CAMP positive, such as Listeria monocytogenes, which exhibits a block-type hemolysis. This differentiates it from other Listeria species, such as Listeria ivanovii. Rhodococcus equi, some Vibrio and Aeromonas species are also CAMP positive, but you do not need to know this for the boards. So while we're on the topic, I would also like to cover the reverse CAMP test, which detects the inhibition or alteration of hemolytic activity. Clostridium perfringens is reverse CAMP positive because it produces an alpha toxin lecithinase that acts synergistically with the CAMP factor produced by Streptococcus agalactiae, which causes enhanced hemolysis. This is mediated by the alpha toxin of Clostridium perfringens, which is a zinc metallophospholipase C enzyme. When this test is performed, Clostridium perfringens will exhibit an arrowhead or bow tie hemolytic pattern. Archaeobacterium hemolyticum is also reverse CAMP positive. So remember that CAMP enhances hemolysis and uses Staph aureus, while reverse CAMP uses Streptococcus agalactiae, and it tends to inhibit or alter hemolytic activity. So here are the CAMP reactions for Streptococcus agalactiae and Listeria monocytogenes, and here is the reverse CAMP reaction for Clostridium perfringens. These are the ones that you'll probably want to know for the boards and for the context of this class. Although Streptococcus pyogenes, also known as Group A streptococci, are one of the primary causes of bacterial-mediated pharyngitis, several other organisms are causative agents as well. Others detectable in a standard throat culture would include Group C and G streptococci as well as Archaeobacterium hemolyticum. Group C and G streptococci are the second most common causes of pharyngitis. The presence of petechiae on the soft palate are pathognomonic for bacterial pharyngitis. Before we dive deeper into some of the beta streps, I want to take some time to talk about Archaeobacterium hemolyticum. This organism is not well defined in terms of normal flora, but it is thought to be a commensal of the skin and the upper respiratory tract. From a morphological standpoint, it is a pleomorphic gram-positive rod that resembles a diphtheroid upon gram stain. On blood agar, colonies are generally small, dull, and matte. This organism is catalase negative, which can help differentiate it from Corynebacterium species that are catalase positive. Clinically, the presentation of Archaeobacterium hemolyticum mimics Group A strep as it produces a scarlatiniform rash. While this organism is not commonly isolated, it is typically found in teenagers and in young adults when isolated. This slide highlights some of the less common causes of bacterial pharyngitis. First, let's talk about Neisseria gonorrhea. So generally, a throat culture is incubated for 24 to 48 hours, but gonorrhea typically requires, 2 to 3 days to appear. Throat cultures usually only use blood agar and SXT, so it's hard to isolate it in time. But Thayer Martin and chocolate agar improve overall isolation of the organism. Patients are often asymptomatic, and this organism is never considered normal flora, so it should always be reported when present. Next, let's talk about Fusobacterium necrophorum. This is especially important in adolescents and in young adults, and it's associated with Lemierre's syndrome, a serious complication involving septic thrombophlebitis of the internal jugular vein. Unlike gonorrhea, this organism can be part of normal flora in some young adults, which makes interpretation trickier. Also remember that recovery usually requires anaerobic culture, so it is outside of the regular workflow for a throat culture. Additionally, we have Haemophilus influenzae. This organism is commonly found as normal nasopharyngeal flora, especially in children, where colonization rates can be exceedingly high. Generally speaking, this organism would not be isolated in a throat culture based on the media present. Policies may vary by laboratory, and some may not report Haemophilus influenzae if present, but some labs will opt to do so, so that way providers can rule out epiglottitis, or the organism can also be significant in immunocompromised patients, such as children that are asplenic, and also persons that have HIV. Here I've listed some of the other causes of pharyngitis. Bordetella pertussis or parapertussis are the causative agents of whooping cough. These organisms are generally not culturable, and they require molecular methods for identification. Mycoplasma pneumoniae is known as walking pneumonia, and it is commonly found in younger adults. This organism lacks a cell wall, and it also requires PCR to be identified. Chlamydophila pneumoniae has a high incidence among elderly populations, and like Mycoplasma, it does not have a cell wall, and this organism also requires PCR. Corynebacterium diphtheriae will be covered in a later module, but this organism is generally not isolated in the clinical laboratory due to vaccination measures in the United States. In the bottom box, you'll see I've listed a number of organisms. These organisms have been implicated in bacterial-mediated pharyngitis, but they may also be considered normal flora or colonization depending on clinical context. This is why organism predominance in a culture is so important in clinical microbiology. The pure growth of an organism in a throat culture should be reported, but it generally reflects a carrier state or drainage from other anatomical sites. Clinical correlation is necessary to determine significance. Lancefield antigens are group-specific C-carbohydrate antigens embedded deep within the peptidoglycan of the cell wall. They are composed of a polyramnose backbone with group-specific side chains for each group. Latex agglutination is used to detect the antigens, and enzymatic extraction using proteolytic enzyme is generally performed because it liberates the Lancefield antigen and increases the overall sensitivity of the test. It is important to note that Lancefield antigen determines the serogroup, but it does not determine species. However, clinically, group A and B are respectively correlated to their species due to strong correlation with respect to their conservative gene clusters and epidemiological predominance in human infections. In the clinical laboratory, we test for groups A, B, C, D, F, and G. So now let's talk about group C and G strep. These streptococci fall out of the scope of a Group A strep culture screen and molecular assays for Group A strep. Clinicians may overlook them as potential causes of pharyngitis, even though they can produce Group A strep-like symptoms, particularly in adolescents and in young adults. Group C and G streptococci are considered commensals of the skin, genitourinary tract, and the gastrointestinal tract. Around 4 to 5% of adults are asymptomatic colonizers in their upper respiratory tract. If Group C or G strep are isolated in a throat culture, they should be worked up because the clinician cultured the throat for a reason. And what I mean by that is they cultured the throat because they're having, upper respiratory symptoms. The most commonly isolated Group C or G strep is Streptococcus dysgalactiae. Although less common, Streptococcus equi and Streptococcus canis are also potential Group C and G streps that are found in the clinical lab that are implicated in pharyngitis. Finally, Streptococcus aeruginosus group, which is normally Group F, can also be C or G. Now let's talk a little bit about Lancefield Group D. These organisms are gamma hemolytic and bile esculin positive. They are generally significant in UTIs, wounds, and bacteremia or endocarditis. This includes both Streptococcus species and Enterococcus species. Enterococcus species used to be considered part of the Group D streptococci, but they are no longer considered a Group D strep. Enterococcus species, such as Enterococcus faecalis, are more often associated with UTIs and wounds than Group D streps such as Streptococcus bovis and Streptococcus galliticus. The enterococci are catalase negative, PYR positive, and they grow in 6.5% NaCl, also known as sodium chloride. The Group D streptococci, Streptococcus bovis and Streptococcus galliticus, are catalase negative, PYR negative, and they fail to grow in 6.5% sodium chloride. It should be noted that Streptococcus galliticus is strongly associated with colorectal cancers. The presence of Streptococcus bovis and Streptococcus galliticus is generally not reported in wound cultures. However, they may be reported if the growth was pure, and there were many whites present in the gram stain. Next, let's take some time to talk about Group F strep. Streptococcus aeruginosus group comprises the majority of the Group F streps isolated in the clinical laboratory. In rare occurrences, some strains of Streptococcus dysgalactiae, subspecies esquissimilis. Group F strep are often implicated in localized purulent infections, such as abscesses. Strep constellatus is more often associated with thoracic intra-abdominal infections with aggressive pulmonary presentations, while Strep intermedius is strongly associated with brain and liver abscesses. Strep intermedius has this CNS tropism due to the presence of intermedullicin and antigen 1/2 PAS, as well as hyaluronidase. Group F strep are normal flora of the upper respiratory tract, gastrointestinal tract, and the anogenital region, and they should never be reported in a routine throat culture. Group F strep exhibit a pinpoint colony morphology on blood agar, and they have a buttery butterscotch odor to them. Lastly, it should be noted that unless isolated from a sterile site, such as blood or cerebrospinal fluid, or in the cases of treatment failure, routine susceptibility testing is not typically required for beta hemolytic streptococci as indicated by CLSI, the Clinical Laboratory and Standards Institute. Penicillin and cephalosporins are the drugs of choice, as resistant to beta-lactams among groups A, B, C, and G streptococci are exceedingly rare. I hope you found this lecture presentation useful. Thank you for watching.